Participant kindly mute your audio.
Dear participants, kindly mute your audio.
வந்துச்சா? இது வந்துச்சா? இந்த வரும். நான் உள்ளே போல. ஐயோ எதான் பாக்குறது நம்ம. எல்லாம் காசு இருந்ததுனால எல்லாரும் வந்துட்டானு. காசு ஆமா. மியூட் இருக்கு, அன்மியூட்னா வெளியே வந்துருமா? நம்ம பேச கேட்கலாம். ஜெராக்ஸா? என்னது? ஜெராக்ஸ் பாருங்க. எங்க இருக்கு? Zoom meet join பண்ணியா?
சார் ஜாயின் ஆக எனக்கு அப்படி நினைக்குது. நீங்க ஜாயின் ஆகிட்டீங்களா? பேஜ் மட்டும் 6 பேஜ் ஓப்பன் ஆயிடுச்சு சார் சாருக்கு. சார் எங்க போனீங்க?
அருண்
அதான் சார்.
வெங்கடேஷ் என்ன ஸ்பெல்லிங்? கே வராத?
ஜி தான் சார் இருந்தது. பாத்துருவோம் சார்.
எந்த உள்ள என்டர் ஆக மாட்டேங்குது. ப்ளீஸ் வெயிட். ப்ளீஸ் வெயிட். ஐயோ எனக்கு இங்க அது வருது.
Dear participants, kindly mute your audio and video.
Okay, thank you. Then Central Council
Dear participants, kindly mute your video and audio. Speak.
ਭਰਾ ਤੂੰ ਵੌਇਸ ਲੱਭਣ ਦੇ ਮੈਨੂੰ ਤਾਂ ਵੀਡੀਓ ਵੀ ਨਹੀਂ ਲੱਭ ਰਹੀ ਆਖਰੀ ਚ ਲੱਗੀ
மீட்டிங் லாகின் பண்ணுங்க. இப்பதாங்க மணி 11 யாரும் பேச ஆரம்பிக்கல. நேத்து அன்னைக்கு வந்து ஒரு மெயில்ல எனக்கு அனுப்பிச்சு விட்டீங்கல்ல, அதே லிங்க், சேம் லிங்க்.
ਕੀ ਲਾਇਆ ਮੈਡਮ
சார் முத்துசெல்வி மேம் கேட்டா உங்க மெயில் இருந்து அனுப்பிச்சிங்க. இதுல இருந்து அனுப்பிச்சு அனுப்பிடுங்க. கரண்ட் போயிட்டாரு, கிளாஸ்ல போயிட்டாரு. வெயிட்டிங் அப்போ அது கனெக்ட் ஆயிடும்.
ਦਰਖਤ ਨੂੰ ਬਿੱਲ ਲੱਗੇ ਹੋਏ ਹਨ
ਬਿੱਲ ਬਿੱਲ ਪੁੱਤਰੀ
ਪਤਾ ਨਹੀਂ ਬਿੱਲ ਬੁੱਲ ਕਹਿੰਦੇ ਹੁੰਦੇ ਇਹਨਾਂ ਨੂੰ ਫਲ ਦੀ
ஐயோ ராமா ஒரு எம்பி எப்போ வரும்? இல்ல இப்ப சின்ன வீடியோ ஆரம்பிக்கல.
फोटो रब कदों पीसी मैं पीसी में देख लेंगे वो है ही नहीं पता आगे कदों आवे मैडम ने आके पता लगूगी इतनी दूर फिर मैंने जाना वो फिर मैं फिर घर जाना है हाँ sure. रुक जाओ मैं क्या फील्ड चाहिए बड़ी मेथी टाइप है वो ਜਿਹੜੇ ਹੁੰਦੇ ਆ ਇਹਦੀ ਭੁਰਜੀ ਬਣਦੀ ਹਾਂ ਬੜੀ ਸਵਾਦ ਹਾਂ ਇੱਥੇ ਲੈ ਨਹੀਂ ਬਣੀ ਹੁੰਦੇ ਤੈਨੂੰ ਆਪਾਂ ਮੈਂ ਲੈ ਜਿਓ ਘਰੇ ਨਹੀਂ ਪਰ ਲੈ ਜਾ ਪਤਾ ਮੈਨੂੰ ਨਹੀਂ ਪਤਾ ਦੇਣਾ ਨਹੀਂ ਆ ਤਾਂ ਬਾਤ ਹੀ ਉੱਗੇ ਪਿਆ
ఏదో కేటాల
Good morning, one and all. I cordially welcome our chief guest, Dr. K. Murugabhupati Raja, Professor and Head, Department of Chemistry, School of Physical Sciences, Central University of Kerala. He has completed his PhD in Molecular Biophysics, Indian Institute of Science, Bangalore. He have done his postdoctoral research fellowship with Professor Dr. Harald Square, Goethe University, Frankfurt am Main, Germany, and also in Singapore and Bangalore. And also he has done his research experiences on 2 ongoing research projects, 8 completed research projects, and 10 international EMBO, FEBS, and other practical courses. He has published more than 100 publications. His PhD thesis is in the topic of design, synthesis, and conformational analysis of peptides containing omega and D amino acids. He has filed US international patent. He has also published 50 journal publications, 5 book and book chapters. It is remarkable that he is a member in Royal Society of Chemistry and also a life member in various societies, including National Magnetic Resonance, Indian Peptide Society, Chemical Research Society. Sir, please.
Yeah, thank you. Can you hear me?
Yes, sir.
Is it clear?
Yes, sir.
Okay. Can you make me host and also allow me to share my screen?
Yes, sir.
I'm not able to screen the share the screen. Host to disable the screen.
Sir, now you can share your screen, sir.
Yes, I'll try.
Dear participants, kindly mute your audio and video.
All right. Can you see my screen? Is it clear?
Sir, it is visible.
Yes, sir.
Okay, great. Okay, first of all, I would like to thank the organizers, especially Dr. Selvaraj, for giving this opportunity to share with some of the interesting aspect of green biotechnology and bioenergy. I am Dr. Murugabhupati Raja, Professor and Head, Department of Chemistry from Central University of Kerala. All right. So I would like to request all the participants to take notes. I'm sure it will be of value to you. Okay, our research earlier I was in Madurai Kamaraj University. Now I'm in Central University of Kerala. Our research center around peptides and peptidoglycans. And since today is on a specific topic, and I'm moving to the straight into business. So here you will see red, white, and green. So today I'm going to tell you something interesting for those, since it is a heterogeneous participant. I would like to really start from scratch. So when it comes to biotechnology, we can actually classify biotechnology into three different perspectives. One is white, red, and green. So white is industrial process and manufacturing of biotechnology. Then anything related to healthcare, pharmaceuticals, we call that biotechnology as red biotechnology. And aspects related to agriculture, livestock management, or all biotechnology related things we call green biotechnology. So I would request all of you to keep your questions for the end. So what is a green biotechnology? So there are in this FDP, there are two aspects. One is green biotechnology, and another is bioenergy. So today I'm going to touch both some fundamentals as well as some aspects of analytical methods. So green biotechnology is, as I told you, anything concerned with this agriculture and livestock management. So some of the examples of green biotechnology are selection and demonstration of plants via micropropagation, designing of transgenic plants to grow under specific environment in presence and absence of chemicals, and these also engineering of plants which can express pesticides so that we don't have to add pesticides additionally. And also biotechnology involves microorganisms to clean and reduce waste. So this is also a very hot area of both research as well as in real applications. And these are some of the applications of green biotechnology. So this is to green biotechnology is useful to in producing renewable energy sources and to improve the plant disease resistance and creating biodiversible or biodegradable materials such as biodegradable plastics, okay, and to enhance the environmental protection and sustainability. What are the different methods of green biotechnology? So hereafter, it's going to be a lot of text. So I want all of you to really make a note because an assessment, you might also find some of these questions from this side. So different methods of green biotechnology include genetic engineering, marker assisted breeding, tissue culture and micropropagation, RNA interface or RNAi technology, and phyto remediation, and bio fortification and precision agriculture, biological pest control, synthetic biology, nanotechnology in agriculture, and also ethical concern. All these comes under different methods, so we can actually apply to green biotechnology. And what is the relationship between the green biotechnology than bioengineering? And also, I would like to emphasize green biotechnology also equivalently used as a plant biotechnology because it is largely based on the agriculture. So from plant, we could actually extract the biofuel, that is production of biofuel, second and third regeneration biofuels, biomass conversion, all the waste, agriculture waste, we can actually convert into biomass, where we can use that biomass to produce energy. And algae bioenergy, biogas production, waste to energy, and metabolic engineering, sustainable feedstocks, and carbon management, and bio refineries, and so on. So I'm going to take some of the aspects and discuss. One is that feedstocks, which has a diverse source of energy. One is biomass, and so biomass, algae, and microorganisms. So biomass, which comprise of plant material, agricultural residues, forestry residues. So it's largely useful in gasification and pyrolysis, basically to produce thermal energy. Then algae is a microscopic aquatic organism with a high lipid content used for biodiesel and other biofuels. And microorganisms, which are engineered microbes for fermentation and microbial fuel cells, as well as bacteria and yeast for bioethanol production. And what are the different processes and products of which should transform raw material into a bioenergy? So one is bioethanol and biodiesel and biogas. These are the products which we get, which is bioethanol essentially from a fermentation of sugars from biomass, uses a motor fuel or fuel additive. Biodiesel is transferication of lipids from plant oils or algae, utilized as a sustainable substitute for the diesel fuel. So similarly for biogas produced from anaerobic digestion of organic materials, which is used for electricity generation and heating. And what are the sustainable practices which largely impact the environment? One is related to carbon, where many of you are already aware that how we can actually manage the carbon sources as importantly involved in climate change and other things. Since today is not focused on that topic, I'm not going to extend more on this. And the second one is sustainable agriculture for the development of energy crops with minimal environmental impact, promoting sustainable farming practices. And third, but very important, is waste utilization. So one of the sustainable, so if you look at the sustainability, so very first thing is reduce and reuse and recycle. So whatever we generate the waste content, then how we could actually first of all reduce, reuse, and also recycle it. So the waste utilization is important source for bioenergy conversion, which will also substantially help in environmental safety. What are the advancements in bioenergy? At least we could classify into 3 levels. One is advanced biofuel, then which is actually utilizing the cutting edge technologies like synthetic biology and algae energy engineering for the next generation biofuels with the improved properties and sustainability. And second is biochemicals, which is deriving from bio-based chemicals from biological feedstocks for industrial applications such as solvents and plastics, biodegradable plastics, and so on. And third one is electricity generation, utilizing the biomass or biogas for power generation. mainly for sustainable energy. And when it comes to this green biotechnology and bioenergy, now we are going to look at what are the different components of bioenergy from green biotechnology. As we told already, we have two components. One is from plants and then from feedstocks. So first we'll look at feedstock, which has biomass, from plant material, agriculture residues, and forestry residues, and so on. And algae, and which is used for biodiesel and microorganism and waste material. And what are the different processes products? Bioethanol, biodiesel, biogas, and syngas or synthetic gas. Then biohydrogen, biochemical, electricity generation, and advanced biofuel. So these are the very hot field, not only for the research and also for the industrial real application of production. And as we already looked into the environmental consideration, other than this carbon and sustainable and waste utilization, biodiversity conservation also very important aspect that we need to take into account. Having said that, what are the key molecules that, because now these are materials, now materials are actually composed of molecules. If you break down into the several molecules, either chemical molecules or biomolecules. So if you ask a question, what are the key molecules associated with this bioenergy? It's going to be a few slides. Continuously, there are I colored in blue, which the different key molecules. One is glucose. The red colored in red is what are the different role and different sources which we can get it from. So glucose, cellulose, hemicellulose, lignin, lipids, triglyceride, ethanol, methane, hydrogen, synthetic gas such as carbon monoxide, hydrogen mixture, then acetate, butanol, acetyl-CoA coenzyme, then pyruvate, then ADP, NADH, and FADH2. So these are about 16 key molecules which actually involve in bioenergy. So I'll again go through glucose, cellulose, hemicellulose, lignin, lipids, triglyceride, ethanol, methane, hydrogen, synthetic gas such as carbon monoxide, hydrogen mixture, acetate, butanol, acetyl-CoA, pyruvate, ATP, NADH, FA. Now, if you look at these molecules, then you can understand largely they are all not very big molecules such as proteins or nucleic acid. They are actually small molecules. And these small molecules, again, based on the chemical nature, they are lipid based and carbohydrate based and nucleic acid based and typical small molecule heterocyclic compounds. Okay, so based on this only, so why we need to know this molecule, because we are going to characterize them and we are going to benefit from based on what kind of molecule we are interested to produce from the biomass or the waste materials and so on, or the from the green biotechnology resources. Okay, then move on to what are the molecules present in bioenergy materials from green biotechnology. So I have classified this into different components. So the first one is molecules from biomass components. So from biomass, largely actually we have cellulose, hemicellulose, and lignin. So they are all polymeric carbohydrates, simple and complex carbohydrates. And the next one is carbohydrates such as starch, sugars, which includes glucose, sucrose, and fructose. They are all very energy-rich carbohydrate, which derive from photosynthesis and microbial fermentation. So there are two components. One is a biomass components, such as cellulose, hemicellulose, lactate, then carbohydrate, and next comes to lipids. So lipids, triglycerides, all these two components from fatty acid related one, highly hydrophobic in nature, which also have a huge energy, so that we can get high energy products such as biofuel. And the next one is proteins and nucleic acids, okay, from which also is involved in enzyme and so on. Enzyme and biochemical intermediates, right? So whatever I said, there are 16 molecules. Now we have classified into the different categories based on the chemical nature. Then short chain fatty acid and gases such as hydrogen, methane, and acetate, pyruvate, they are short chain fatty acids. Okay. Then acids and secondary metabolites, because when a biochemical reactions happens in the cell, and finally it produces a lot of metabolites, mainly secondary metabolites, such as terpenes, phenolic compounds, and so on, in addition to the acetic acid, which is also one of the very important metabolic intermediate, okay, and also produced during the fermentation and bioconversion. Okay, so this is all about molecules. Okay, so for now, what we have seen is different processes, some of the components, so that I will take everyone into a same level. And based on these different processes, green biotechnology processes and resources, we will have several molecules which extracted or which we can get it from biomaterial. Basically about 16 plus molecules, key molecules. Now we move on to what are the different analytical methods which we can use in green biotechnology. So when it comes to green biotechnology, then biotechnology involves larger molecules or larger biomolecules such as protein, nucleic acids, and so on. So that means we are going to manipulate, or we are going to. Could you please, accept the host and host, all others? Can you please mute your mic? Thank you. Okay, so these are the host. Can you please? Yeah, thank you. So DNA sequencing. So here important bioanalytical methods for which we use in green biotechnology, like DNA sequencing, PCR, polymerase chain reaction, and proteomics and metabolomics. Then fluorescent microscope, flow cytometry, enzyme-linked immunoassay like ELISA, then next generation sequencing. These are the very important analytical method which we used in green biotechnology. Okay. In what are the, this is 8 methods, or these are the 8 methods which we use in green biotechnology. What about analytical method used to characterize bioenergy? I mean, molecule which produce or material which produce bioenergy. So, following the different analytical methods, which are entirely different from which we use it for biotechnology purpose or characterization. largely chemical based, because here we are going to characterize small molecules which are the products or resources for the bioenergy production. One is biomass composition analysis to determine cellulose, hemicellulose, lignin content, and optimize biomass for fuel production. Then gas chromatography and mass spectrometry, that is GCMS, which separates and identify volatile compound and gas molecules, okay, and also analyze biofuel components and fermentation byproduct. And third one is nuclear magnetic resonance spectroscopy, where we can analyze the molecular structure and of the bioenergy molecules or resources. And this also provides insight into metabolic pathways, different intermediate, and how this would have been reached. And the fourth one is high performance liquids chromatography, or HPLC, which also largely used to separate and quantify compounds in the liquid cell. So GCMS for the molecules which are volatile, which largely used for the gas phase molecule, and HPLC used for the liquid state molecule, a sample which is in the liquid state. And also, very importantly, it is useful in quantifying the sugars, and also qualitatively, we could also from qualitatively characterize and also quantitatively measure the amount and other components. Okay. The fifth one is biochemical assays. There are a lot of biochemical assays, like such as quantity to quantify the enzyme kinetics, enzymatic assays, and monitors the key enzyme influence biofuel production and so on. And the sixth one is thermogrammetric analysis, or TGA. This would tell, importantly, the weight change with respect to different temperature when we actually convert this biomass into a energy converter. So it basically we actually consume them or burn them. Then with respect to the heat, what is the change in mass? And also we can study the thermal properties of biomass for energy conversion. Then microbial fuel cell analysis, which will give us electric output from microbial fuel cells. Essentially, one can actually measure what is the potential use of these bioenergy molecules or materials, which can be converted into the electricity. Then the last one is life cycle assessment, LCA, which evaluate environmental impact of bioenergy production and essential for assessing the sustainability in bioenergy process, mainly for a regulatory purpose. Okay. Then I'm going to tell you, so these are the 8 analytical methods in bioenergy. So today I'm going to pick up few, and I'm going to tell you how this could be utilized in order to characterize materials which gives bioenergy. In the since we have a very limited time, I'm going to only give an overview. If anyone want to know more about it, we can discuss it in the question answer or some other time. All right. So now we have a material. Now we want to characterize this one. So there are several methods. So one of the, or largely they are all very similar. I just picked up one workflow methods where we have a sample. Then we need to first, because all the biomaterials have more than several molecules together. Because any material, they are going to be heterogeneous, both in molecular nature as well as the properties. So first we need to actually isolate them in order to identify what are the components present in the material, as well as we need to actually quantify it. So first thing is we need to extract, then separate, then characterize. Largely we can characterize by mass spectrometry and spectroscopic methods. So when it comes to any molecule or material, there are only few ways we can actually characterize. One is based on the mass. then of mass of the molecule, which is very unique. However, with the same molecular weight, we can also have several similar molecule. So we need to actually characterize their structure and properties. So we will be using separation and spectroscopic methods in order to characterize their structures, functional groups, and so on. And the third one is with respect to the energy or with respect to the temperature, how much mass is lost, like TGA or DSC, so on. Okay. All right. So I'm going to pick. I'm going to tell one each. So, so the very first thing is GCMS. Some of you would have been already aware of it. So GCMS is gas chromatography and mass spectrometry. So which largely we use for only volatile compounds, volatile molecules. Okay. So this is a typical GCMS or GC or so we have two components. One is gas chromatography, which is used to separate these different components, volatile compounds in the mixture. Then another one is to identify what are the molecular weight of each separated components or compounds. So there are you can actually use them standalone, gas chromatography, or you can actually combine them both as well. Okay. So largely, if you combine them, we'll have a GCMS, which are very, very useful to characterize precisely what is the molecule. So what are the advantages of gas chromatography and mass spectrometry or GCMS, which are highly sensitive? We could actually pick up precisely based on the RF value and the mass. We can actually identify even with a very low concentration present in this one. So we can use it for chemical identification and also used for quantitative analysis. So once we have a established molecule, then we can make a calibration curve, then we can actually measure it or quantify it, how much exactly present in the even unknown sample or material. And how we could actually do this experiment or GCMS, there is a typical workflow. So first thing is we need to prepare a sample because it need to be dissolved in the right solvent. Then we need to choose a right column where this separation can be best. And then we need to also choose a suitable analyzer in the mass spectrometry. There are several analyzers available. So sample preparation, then chromatographic separation and mass spectral analysis. Once we have a mass spectrum recorded, then we can actually analyze it based on because these are very simple molecules or small molecule, which are well established, so which can be easily characterized. Then we'll go to the spectroscopic techniques. There are several spectroscopic techniques. Most of you, those who are in biotechnology science research, already you will be definitely aware, and also many of you would have been already used. So you would have used the UV visible spectrometer, then atomic absorption spectrometer, Fourier transformation infrared spectrometer, Raman or FT, NMR, X-ray spectroscopy or X-ray crystallography. So X-ray spectroscopy for the elemental analysis, which we use seldom in bioenergy because largely they are carbon compounds. So we don't use these two, but other spectrometers, we use it to characterize structure and also for quantification and functional group analysis. So I'm going to throw some light on some aspect of FTIR spectroscopy. So basically, FTIR spectroscopy to identify the functional group. So these are the principles. So when we have two atoms connected with the bonds will move or move their position, internuclear distance will change. So we call this is a vibration, stretching or bending and so on. So this can be measured based on the energy you supply. So this can be identified. So we will have several motions of these bonds, such as symmetric, asymmetric stretchings, and bending, plane, in plane, out, wag, plane, out of plane wagging, and so on. So this functional group, motions can be measured in the infrared region of the uh, electromagnetic radiation. Okay. So what are the different applications of vibrational spectroscopy? So vibrational spectroscopy, again, we can, there are two spectroscopy. One is FTIR, another is Raman. So there are a different type of FTIR, ATR, 2D, vibrational circular dichroism, time resolved, and so on. Depends on the specific. But for bioenergy materials, we will most of the time we will use simple FTIR, where we are interested only in the stretching frequency. Okay. One can also use vibrational imaging microscope. It's a very advanced one, but we don't do that. So there are FTIR spectroscopy is a quite versatile spectroscopy because it's potential to use this as a in a different molecules and their different environment. So whether it can be liquid, solid, even gas, gel, membrane, with a different concentration, medium, temperature, pH, all can be used FTIR spectroscopy, unlike other spectroscopy, which are have some limitation. And also we can characterize biomolecules and typical small molecules, functional group of organic molecules, metal ions, and also useful in mechanistic isolation and molecular interactions. Okay, this is the typical FTIR spectrometer. If you are open it, that's what you will see it. But today I'm not going to explain more on this. So we have a source and beam splitter. Then we have a interferometer, then a detector. Okay, so based on the choice of this one, then we will have a mid IR, that is what largely used. Then we have a far IR and we have a near IR spectrometers, which all have a different. But for most of the organic type of molecules or a small molecule, which we extract from the bioenergy materials, generally we use it for mid IR spectrum. So typically, there are a few parameters that we need to concern about: the spectral resolution, whether it is a continuous scan, and so on. But now most of the spectrometer, when they all set to directly make a measure. It's very simple measurement. But what we need to pay attention is sample preparation, whether your sample is liquid or solid or a gas. Based on this one, then we need to actually sample preparation. And importantly, after once you collect the data and analyzing the spectrum, it's actually quite challenging because IR spectroscopy is a quite complex one because extremely difficult to interpret all the peak, unlike in NMR spectroscopy or mass spectral data. Okay. And these are the merits and challenges of IR spectroscopy, but largely we can actually use it. But when we come to the complex spectrum, we could also get advantage of really resolving this overlap peak by applying the mathematical functions in order to really separate this overlap peak, such as FSD or derivative method and so on. And the next one is a nuclear magnetic resonance, which is another very important analytical method largely used in order to identify the structure of this molecule. Okay, so many of you already are aware this is not only expensive one, but also it needs a lot of attention to interpret the data and the sample preparation and so on. Okay. So the role of NMR spectroscopy is for structural analysis, metabolic profiling, and biomass characterization. So largely we will use NMR spectroscopy to identify, based on the chemical shift value, we can actually identify what type of molecules are present. So very importantly, we could use any of these spectroscopic method only for the molecule which are pure. Otherwise, it makes no sense, because once you have a direct biomass, many of researcher unknowingly or with the less awareness, they directly use as a crude material and trying to characterize. It is a very wrong approach, and also you will not get. Instead, you will actually adding confusion to the problem which already exists. So the very first thing is you need to separate them, either using gas chromatography, GC, or using the HPLC. If they are liquid, then you have to separate them, then characterize this homogeneously pure molecule. Okay. So NMR spectroscopy, it's also used for quantitative analysis, structural elucidation, and importantly, process optimization. Okay. And the third method, which I'm going to explain, or fourth method, is actually thermogravimetric analysis of bioenergy material. It's a very vital tool, largely used to assess the worth or the potential of the biomaterial as a energy material. So once it is so today is I do not have much time to really explain all the TGA, but however, once we get a TGA, so what we are essentially doing is we change the temperature and keep the material and measure that loss of mass, okay, with the every time, every temperature point, okay. Then one can actually identify quantitatively, and we could also make a devolatilization profile. Then we can also evaluate the thermal stability of the material, okay, whether it is suitable for the bioenergy application or not. So TGA is used in bioenergy feedback evaluation and emission reduction studies and characterizing biochar. Okay. All right. So what are the key parameters used in TGA? One is, as I told you, weight loss, then derivative weight, and residue analysis. These are the three important parameters which used in TGA. Some of you already also aware there is also another method, the thermogravimetric analysis, and we also have a very equivalent one is DSC, that is differential scanning calorimeter. So in differential scanning calorimeter, which we measure the heat flow into or out of the sample as a function of temperature or time. Whereas in TGA, we measure the weight change as a function of temperature under controlled conditions. So this is a very important difference, which difference between TGA and DSC. Okay, so what are the future purpose perspective of TGA is for advanced characterization, and one can also actually make a high throughput analysis by making it automation and robotics and multi-component analysis and so on. Okay. However, every method has a limitation. So what is the challenges in TGA? So the sample.
Excuse me.
Yes.
Sir, excuse me. Requesting all other participants to mute your video and audio, please. There are some disturbances. All other participants, kindly mute your video and audio. Dr. Selva Kumar, please. Mr. Vishwa, please. So there are some lag in streaming due to participants are switching on their video.
Yeah.
Sir, please mute your video, sir. Dr. Rajasekaran Yelumalai, Dr. Balasubramanian, kindly mute your videos. Dr. Balasubramani, Kindly mute your video, sir. Dr. Rajashekaran, kindly mute your video. Dr. Majid, please mute your video. Stop your video, please. Mr. Victor. Yeah, if everybody switching on the video, there is some lag in the streaming. Please switch off your video, please.
Yeah, host can actually mute everyone's video except the co-host and host.
Yes, sir.
That
Yes,
can be.
sir.
Yeah.
Yes, sir. I'll do that now. Thank you, sir. Thank you.
Yeah, thank you. Okay, what is the challenges in TGA? So it's importantly 4 challenges. One is sample homogeneity, calibration accuracy, and atmospheric interference, because we are actually changing the temperature. So atmospheric, so we need to also take care of that one. And importantly, how we could actually interpret the data. So these are the challenges while we are actually analyzing the TGA results of a bioenergy materials. All right, so far what we have also quickly looked at 4 or 5 methods. One is GCMS, then FTIR, I told about, then NMR spectroscopy, then TGA. Okay, 2 spectroscopy, one for characterization, another for functional group, and one for mass and separation, GCMS and TGA. There are other things, but in the interest of time, I could not cover, because each topic itself separate, it takes a lot of time. And I will move to the last part of my presentation, that is, what are the innovations in bioenergy? So, which we can categorize into 3 parts, like research and development, where continuous innovation research are shaping the future of bioenergy and green biotechnology. A lot of R&D work is going on in the bioenergy sector, both government funded as well as private funded, because huge hope is in front of us, and emerging opportunities which bioenergy presents new opportunities, creating impactful and sustainable energy practices. Then sustainability focus. Now, green biotechnology is almost a central role in sustainability of environment-friendly solutions, okay, towards the industry or biotechnology industry. And what are the future prospects of bioenergy revolution? Okay. So as I told you already, that research and development, sustainability, which actually brings a global impact, reshaping the global energy industry with sustainable alternatives. So I hope I have given some overview of bioenergy, materials, and green biotechnology, how we could actually use overall, what are the different aspects of analytical methods, and few also I explain a bit. Okay, so if you have any questions, we could actually discuss. Again, thank you very much for your kind attention. Now it's open for questions. Or you can put the questions in chat box.
It's time for query sessions. Dear participants, is there any queries?
My name is Asohan from Mahatma Engineering College.
Okay.
So what is the primary plant growth through the soil is essential? What are the ingredients for development of the plant to be growth?
Okay, actually, I'm not expert in that one. You should really ask somebody having an agriculture background. Okay. I'm a chemistry background, so I'm good at analysis of biomolecules and organic molecules characterization. But for soil, basically, again, nutrients, and it should give a conducive environment for the better growth, nutrition, and it should be protect from the pests and other things.
On the half of the roots of the planets, how to grow inside the soil, how to develop that the roots length to be increased to grow the planet.
Okay, so what's your background, sir?
Associate Professor Mechanical.
Okay, mechanical. Okay. Actually, I'm not expert, so I'm afraid that I will be the right person to answer your question. So maybe the coordinator can help to find suitable resource person to address your question, sir. Thank you, sir.
Yes, yeah. I'm in the way of find out the other planet.
Okay.
Growth the
Okay.
plant
Interesting.
on this area.
Okay. Great. Great.
On the soil test content, which one is required? On the soil test, which one is required to grow the planet?
You mean
The planet.
okay, conditions. Yeah. Mainly for growing anything, we need water, that means moisture, then air, oxygen. So basically the plant has to do a photosynthesis, okay, to produce more.
Okay. From
Yeah.
the sunlight.
Yes. So it should have a chlorophyll. For all the things
Chlorophyll.
basically you need a plant which you should be able to supply water, oxygen.
Yes, maybe.
Yeah.
This one also passing the clouds through the area to reach the planet.
Correct.
This is another one concept we are doing that.
Yeah.
Reach the clouds with the water molecule.
Yes.
Reach the planet. And then what is the condition required for the soil to grow?
Yes, sir.
The planet. What is
I
the
think
requirements?
NASA is already doing a lot of research. You can actually, there are many articles as, I don't know how many, but there are quite interest groups are working on this.
But one thing, sir, sir, hello?
Yes.
Yes. Sir, nowadays we are testing that in moon and the another one planet, red planet, the white.
Yes.
This is going on. What is the content of the soil in between these two?
I don't know, sir, because this is geology and meteorology there, but it is documented. There are quite a bit.
Can you give any notes about that? The planet, which one content more in the soil between the Savoy and Love?
Yeah, many things actually you could also straight away get it from Google, sir. It will be available. Now it is accessible.
Yes, sir. Thank you.
You can try, sir. Thank you, sir.
Thank you.
So there is a question, what type of molecules out of the ones described in your slides can thermometric gravimetric analysis can be done? All right. So any molecules which have a high melting point and material characteristics such as cellulose, any polymeric material, fatty acids, all the things we can in biomass can be used straight away. So for TGA, we can use material as such. For molecular characterization using spectroscopy, we need a molecule which is separated and purified in purified compound.
Thank you, sir. Thank you very much, sir. Thank you. I ask the mic. Okay, sir.
Sir, good afternoon. Hello.
Yes. Yeah.
Hello.
Yes.
Sir, actually, when you are talking about the bioenergy or the lot of biomass is available in our country, but the conversion is very, very small. I think the contribution of bioenergy to the total energy potential is very, very low, I think.
Yeah.
Because the better methods, whether we are unable to find better methods for one method is actually usually we are briquetting it and using the biomass as a fuel. Otherwise, we are going for gasifications. But why so much of lagging in this area actually, particularly in the bioenergy? We can't see much of bioenergy plants in the country.
The first thing is we have not developed many technologies which are cost effective, and also because we spend more time to get less output. That's why many people are not interested, or it's still not translated very well. Okay. And also awareness, basically awareness and regulations. But now most of the Western countries also actually, even it is, so for example, plastic bags are very cheap. However, anything which is biodegradable bags are expensive. So how much time we are really ready to pay that much extra money to actually having an environmental friendly, right? So similarly, for this energy conversion, everything, it is actually better than Esther past, but we will be surely do much better in the future, because now technology is developing. There are a lot of push from the government and also NGOs. A lot of awareness is there. It's coming up.
Thank you, sir.
Okay, any other questions?
Thank you so much, sir, for your informative presentation. Let's wind up the session.
Okay, thank you very much.